Scientists claim to have finished the whole genetic blueprint for human existence, completing a puzzle that was nearly solved 2 decades ago. In a study published on March 31 in the journal Science, a multinational team detailed the first-ever sequencing of a whole human genome, the set of instructions for building and maintaining a human person. The earlier attempt, which was widely lauded across the world, was incomplete because DNA sequencing tools at the time couldn't read crucial bits of it. Even after upgrades, it still lacked around 8% of the genome. Some of the genes that make us distinctively human were actually in this dark stuff of the genome, and they were completely ignored, said Evan Eichler, a University of Washington researcher who participated in both the current endeavor and the initial Human Genome Project. It took more than 20 years, but we eventually finished it. Many people, including Eichler's own students, assumed it had already been completed. I was instructing them when they said, "Wait a minute. Isn't this like the 6th time you've declared victory?" I said, "No, this time we really, truly did it." According to scientists, this complete image of the genome will provide mankind with a better knowledge of our evolution and biology while also paving the way for medicinal breakthroughs in areas such as aging, neurological diseases, cancer, and heart disease. We're merely increasing our chances to study human illness, Karen Miga, one of the 6 studies released on March 31, said. The study is the culmination of decades of labor. Leaders of 2 competing groups, an international publicly sponsored initiative led by an agency of the United States National Institutes of Health and a commercial corporation, Maryland-based Celera Genomics, unveiled the first draft of the human genome in a White House event in 2000. The human genome is composed of around 3.1 billion DNA subunits, which are pairs of chemical bases denoted by the letters A, C, G, and T. Genes are strings of these lettered pairings that carry instructions for producing proteins, which are the building blocks of life. Humans contain around 30,000 genes, which are grouped into 23 groupings known as chromosomes and are present in the nucleus of every cell. Previously, there were huge and persistent gaps in our map, and these gaps fall in rather crucial places, Miga explained. Miga, a genomics researcher at the University of California, Santa Cruz, collaborated with Adam Philippi of the National Human Genome Research Institute to arrange a team of scientists to begin from scratch with a fresh genome with the goal of sequencing it all, including previously missing sections. The telomere-to-telomere, or T2T, consortium is named after the portions of chromosomes called telomeres. Their research adds new genetic information to the human genome, corrects prior errors, and unveils large regions of DNA that have been linked to both evolution and illness. Before being vetted by scientific colleagues, a version of the study was released last year. This is a significant advance, I would say, of the Human Genome Project, geneticist Ting Wang of Washington University School of Medicine in St. Louis, who was not involved in the research, said. According to Eichler, some experts used to believe that undiscovered parts contained garbage. He's not the one. Some of us had always thought there was gold in those hills, he explained. According to him, gold contains many critical genes, such as those involved in making a person's brain larger than a chimp's, with more neurons and connections. Thank you for watching. Please like the video, share and subscribe.